Okay, this is Mr. Maxwell, and this is a screencast largely for those of you who had to miss class for one reason or another. This one is much better done in class, but if you pause and think about the questions I'm asking you, you'll probably get a fair amount out of it. You might just run through it if you've already seen this in class, but at any rate. So once upon a time, we didn't know what was the genetic material, and this was one of the first experiments, and it was done by a guy named Griffith, who was looking at some cool things. He noticed basically there are two different types of bacteria he was looking at. There's smooth bacteria in a colony, and then there are bacteria that look rough in a colony. And there are two types of mice, living mice and dead mice. And so the first part of this experiment, you take smooth bacteria and you inject it into a live mouse, and you get a dead mouse. And if you take rough bacteria and inject it into a live mouse, you get a live mouse. So you ought to be able to tell, what's the information that this experiment gives you. So stop it, think about it, write it down. So it ought to tell you that smooth bacteria kill mice and are therefore pathogenic. Rough bacteria don't kill mice. Now you can do a follow-up study here where you can re-isolate bacteria from the mouse, and you're going to find that you get smooth bacteria out of this mouse, and you don't get any bacteria out of this mouse. So the next experiment then, you take the bacteria and you boil them for 10 minutes so that you can kill them. If you boil them and try to grow them on LB, they won't grow because they're dead. So you take a live mouse and you boil bacteria so that you get dead, smooth bacteria, inject it in the mouse lives. Again, then, you take a live mouse, inject it with boiled, rough bacteria, and the mouse lives. So, stop the tape, stop the recording, and what does this tell you? Answer that question. So you should have decided that basically boiling kills the bacteria, and you have to have a live bacteria to kill a mouse. Shouldn't be surprising to you, but that's the way it is. So the third part of this is pretty cool. You take a live mouse, and in the syringe you put dead boiled smooth bacteria, which, remember, live smooth cause disease, cause death, and live rough bacteria. And so when you put the dead smooth and the live rough together, you get a dead mouse. Likewise, you can do the other experiment where you take the mouse and put dead rough bacteria and live smooth bacteria, and you know that the mouse dies. So stop, think about why, and write something down. So hopefully you decided that, at least in this case, the reason that this mouse dies is because the live smooth bacteria kill it, and the rough bacteria are basically not doing anything. And then here, hopefully you've concluded that this live rough bacteria must have taken something from this dead smooth bacteria such that they're able to kill this mouse. Now, you should be able to think up of follow-up experiments that you could do to figure out why, and I think you should stop and think about what those should be before I tell you what they should be. So, hopefully, one of the things you might have come up with is, well, let's see what kind of bacteria we get if we isolate bacteria from this mouse. And what you wind up with is live smooth bacteria. And so when Griffith did this, he concluded that there's some factor in this dead smooth bacteria that the live rough bacteria took up that basically transformed this rough bacterium into a smooth live smooth bacteria, and that allowed them to kill the mouse. Okay, so to follow up from this, then, the question is, so what is actually the thing that's doing the killing? So Avery, MacLeod McCarty followed up on Griffith's work. They took heat-killed S cells. Remember, these are smooth bacteria. These are the ones that kill mice. And they boiled them. And then they did a procedure where they removed the lipids, they removed the carbohydrates, because it was already pretty well known that neither lipids nor carbohydrates contain the genetic material. And so then to this tube they added proteinases, and of course protein aces chop up proteins. This sample shouldn't contain any protein. And then to another sample they put RNases, ribonucleases, and so that should degrade the RNA. And then to this third one they added DNases, and so that should contain no DNA. And then they added live R cells and looked at, do we get S cells? Do we get transformation? And in the tube where they degraded protein, they got transformation. In the tube where they degraded RNA, they got transformation. In the tube where they degraded DNA, they got no transformation. And so this was pretty good conclusive evidence experimentally that, if you knock out DNA and you can't get transformation, that DNA must therefore be the genetic material. Think about it for a second. It's a beautiful experimental design. Unfortunately, it really wasn't enough to convince everybody. Another experiment that was done with phage, and phage are basically bacterial viruses, so viruses that infect bacteria. And people knew at the time that phage were composed of protein and DNA. And so what they did was figured out a way to label the DNA and label the protein, reasoning that if you can figure out what gets inside the bacteria, causing it to change from a normal bacteria to a virus production factory, that you could figure out what the genetic material was. And so in one mix they used radioactive sulfur, and in another mix they used radioactive phosphorus or phosphate. And as you know, sulfur is a component of proteins because cysteine contains sulfur. It's an amino acid that contains sulfur, and there's no sulfur in DNA. On the other hand, DNA has a sugar phosphate backbone, and so there's phosphate in DNA, whereas there's not phosphate really in proteins. So the procedure here basically is you mix the two different things so that you can radioactively label the protein, and in another tube you radioactively label the DNA. And so here we've radioactively labeled the protein, and we did that by the sulfur. And when you allow the phage that are labeled to mix with the bacteria, you blend them up, chop them up, and that causes the particles to fall off. And then you centrifuge them to. When you centrifuge, you spin really fast, and the heavy stuff goes to the bottom. And then you look for where does the radioactivity go. Well, the cells are going to wind up in this pellet here, and the supernatant's going to contain the empty protein shells, because the bacteria are a lot heavier than the capsid protein of the phage. And so when you label the protein, you wind up with radioactivity in the supernatant, in the liquid, and none really in the pellet. If you radioactively label the DNA, The DNA winds up being radioactive, of course, and because the DNA goes into the cell, into the bacterium, the radioactivity winds up in the pellet where the cells are. So because the radioactivity is in the pellet, you know that the DNA went into the bacterium, and therefore it's the DNA that must be the thing that's transforming the bacteria from a normal bacterium into a virus production factory. So that wasn't even enough, though. It wasn't until Watson and Crick discovered the structure, or figured out the structure of DNA, that people really bought that that DNA was the genetic material. And there's a really good book called The Race for the Double Helix. There's a movie out for it too. Very good, worth taking some time to see. That whole the whole history of that's really kind of interesting. But I'm not here to teach you history.